All right, you guys, 12:50, let's go ahead and get started. I am recording this lecture. There's a lot of people that are on field trips today, so this one will be posted along with the review session today too. So don't forget the review session is today between 2 and 4 in this room, okay? And I have another 20 minutes before we start the review session. If you have any questions about the problem sets that you're going to be turning in, right? Today, the respiratory essential calculations are due and into thin air. So if you have any last minute questions, I can answer that before we turn those in today, okay? All right, so let's see here. I am recording, and let's go ahead and start with the lecture today. Okay, so what we're going to do is we're going to start lecture 15. If you guys have had a chance to open that up. And the topic today is transport of gases from the lungs to the blood, right? And then transport of those oxygen molecules to the tissue. And we'll be talking about hemoglobin today and hemoglobin dissociation curves. These curves can be very confusing. So I'm going to go through it fairly slowly, and then I'm going to give you some tricks as to how to remember how to think about those curves. Okay, so let's start off with what are some of the normal gas pressures. Last time we talked about partial pressures and how to calculate partial pressure. Remember that it is the atmospheric pressure times the percentage of the gas, of a particular gas, in normal air. All right, so when we look at this, I want to first concentrate on the partial pressure of oxygen. It's really giving you some idea of the levels of oxygen in different parts of the circulatory system. So the venous blood has about 40 millimeters of mercury. Arterial blood is about 100 millimeters of mercury, okay? Alveoli about 105, and the atmosphere, it's 160. So that is quite a dilution effect. If the atmospheric partial pressure of oxygen is 160, it really is diluted, right? Remember, fresh air is going to mix with stale air. In the alveoli, it's only about 105. Then there's not much of a drop from the alveoli to the arterial blood, but then after it passes through the tissue, it drops from 100 to 40. Now, the partial pressure of carbon dioxide toggles between 46 millimeters of mercury and 40, okay? From the venous blood, it basically diffuses into the alveoli and then out into the atmosphere. So remember, you get rid of that extra CO2 just by exhaling. Okay, so do kind of be familiar with some of the normal gas pressures. This chart is actually just giving you an idea of how to think about the partial pressure of oxygen in the pulmonary capillaries and how that's affected by capillary length. So with a normal person who doesn't have diseased lungs, you can see it jumps up from 40 millimeters of mercury. That's what it is in the veins, right? Systemic venous pressure. It jumps up to 100 to 105 millimeters of mercury in the first, I would say, 30% of the capillary length. That's in a normal individual. However, if there's someone that has COPD or a serious lung issue, you can see that it takes the entire capillary length just to only get partial levels compared to a healthy lung. Okay, so COPD is a good example of this. Or pneumonia. There's a lot of different diseases that would cause this curve to be shifted. So again, this is just a nice graph that's showing you the gas exchange between alveoli and blood. All right. Now this is something called ventilation perfusion matching. Okay, it's the matching of ventilation and blood flow in the alveoli. It's very interesting that your body has these mechanisms to match healthy alveoli that's being perfused with or being ventilated with air and healthy blood supply, right, that is going to surround each of these alveoli. So if you actually will take the left-hand side first, if you have decreased airflow to regions of the lung, you really don't want to perfuse it with blood if there's not going to be any gas exchange. So to minimize that, what happens is there's a sensor that detects the lower oxygen levels and it constricts pulmonary vessels to decrease blood flow to diseased areas, to diseased alveoli, right? Why send blood to alveoli that's not getting any air? That doesn't make any sense. So what it does is it diverts blood from areas of disease to areas of healthy air, healthy lungs. On the right-hand side, you can see that the same thing happens if you have decreased blood flow to certain regions of the lung. Right? With decreased blood flow, you're going to get in the alveoli decrease in the partial pressure of CO2, which is going to cause bronchoconstriction. Again, that's going to help you match the healthy areas of blood flow with healthy areas of the lung. Local ventilation decrease to match a local decrease in perfusion. All right, this is called ventilation perfusion matching. And again, the whole idea here is to divert blood flow and airflow away from areas of disease to areas of a healthy lung. Any questions about that, you guys? Kind of an interesting idea. Now, because of this, right, in your into thin air, this is one of the causes of HAPE, high altitude pulmonary edema, this matching. So a lot of times if you're at high altitude and you're not getting, the normal levels of oxygen in the alveoli, it may constrict blood flow and cause edema. All right. So here's another concept that I want you guys to know. This is nice that it has these little balloons here. It lets you know what's going on here. In environments that have low oxygen levels, there's a mechanism in place that helps upregulate proteins like hemoglobin. So this is called HIF1. It's hypoxia. Hypoxia is environments of very low oxygen. Hypoxia inducible factor 1. Now, normally in normal oxygen, this alpha subunit usually gets broken down. But under conditions where you have hypoxia, hypoxia again is an environment that has very low oxygen, it doesn't get broken down and it actually binds to another subunit, an alpha, and the alpha binds to the beta, which helps translocate it from the cytoplasm into the nucleus, where it then initiates protein synthesis of hypoxia response elements, like hemoglobin. So it makes sense that it would be a sensor. It would detect the low oxygen environment, and it would upregulate proteins like hemoglobin. So this is kind of an interesting idea. When I used to oversee the Animal Diversity Lab in CBS, we used to use Daphnia, these little water bugs, to feed the hydra. This is another animal that is closely related to jellies. And it was interesting, we'd have these little water bugs, these Daphnia, in a petri dish all day. And in the morning, they'd be on, the environment would be great. They'd have high oxygen levels. And then over the course of the day, they would use up all the oxygen in these little petri dishes. And the bugs, the little water bugs, would start to turn pink. They were literally upregulating hemoglobin. So you could actually see it without even a microscope. They would start changing colors. And that's because of the action of HIF, hypoxia-inducible factor. Okay, so let's, talk about hemoglobin. Let's start talking about the transport of oxygen in the blood. This is a nice picture kind of giving you an idea of the heme group that is associated with hemoglobin. It's this iron core where oxygen binds to the iron so that it can be transported throughout the body. And this heme group is located in the hemoglobin molecule. So we'll talk about hemoglobin in just a second. Here's the heme group. Myoglobin is a single subunit that has only one heme group. Whales have this, a lot of myoglobin. It gives them the ability to dive really deeply for, hours at a time. That's why this is featuring the whale myoglobin. I'll blow this up a little bit. So you can actually see the heme group, and only one oxygen molecule can bind to each of these heme groups. So a myoglobin molecule can only bind one oxygen molecule. But whales tend to have lots and lots of myoglobin. In humans, the myoglobin is actually within the muscle cells. Now, in the blood, we actually have what's called hemoglobin. Okay, and it is made up of, it's a tetramer that's made up of 4 hemoglobin subunits, 2 beta in adults and 2 alpha. And each of these, you can see these little discs here. These are the heme groups that I just talked about. And each of those heme groups, they carry one oxygen molecule. Okay, any questions so far about the anatomy of a hemoglobin? Okay, so you've probably seen this before. Just want to make sure. All right, so how does hemoglobin, now that you know something about partial pressures, how does hemoglobin affect partial pressures? Now remember, partial pressures are giving you an idea of what the oxygen levels are in a particular liquid. Okay, so it's the soluble oxygen. In this case, what you see is 2 compartments. It's pure water with oxygen within it. Okay, so in this case, they have already reached diffusional equilibrium, right? So the partial pressure of oxygen are equal in both of these compartments. Now, if you actually drop in a hemoglobin molecule, right? This HB is hemoglobin. Now some of the oxygen is going to bind specifically to the hemoglobin molecule and lower the partial pressure of oxygen in compartment B. Does that make sense to you guys? So now you have less oxygen because now some of the oxygen are attached to hemoglobin and it doesn't now factor into the partial pressure within that liquid, within that water sample. So now diffusion of oxygen is going to be from high concentration, compartment A, to low concentration in compartment B until a new equilibrium occurs, right? So this would be a new diffusional equilibrium. So you can see in compartment B, you can carry a lot more oxygen when hemoglobin is present. Yeah.
How many oxygen constants inside do you have?
4. So, yeah, that's not a really good one. Good. Nobody's ever said that before. That diagram is a little misleading. Yeah, absolutely. So there should be only 4 oxygen molecules for each hemoglobin. Yeah, so 6 doesn't make sense. Good catch. Okay, so the take-home message here is adding hemoglobin to compartment B substantially increases the total amount of oxygen in that compartment, since the bound oxygen is no longer part of the diffusional equilibrium. Really important concept. Okay, one more thing, I just want to mention this before we talk about oxygen dissociation curves, is this will come into play when I finish talking about DPG. Okay, that makes no sense to you right now, but it will. Just know that there is a real difference between fetal hemoglobin and adult or postnatal hemoglobin. You can see right after birth, that's when an organism, a human, actually this applies to all mammals, it's upregulated, the beta globin is upregulated right after birth. So remember, hemoglobin in babies and adults have 2 alpha globin and 2 beta globin. But a fetal hemoglobin has 2 alpha and 2 gamma globin. And you can see the gamma globin wanes, really decreases right after birth. Okay, so 2 alpha and 2 gamma fetal hemoglobin. So this is really going to come into play after I talk about DPG. Okay, so this is what an oxygen dissociation curve looks like. Now, this gives students a lot of trouble. I'm just saying this isn't very intuitive, and I want to go through it very deliberately so that you guys can, I'm not going to assume anything. I'm just going to go through it to make sure that you understand this graph backwards and forwards. So the x-axis here is giving you some information about oxygen levels, the PO2. Now this is dissolved oxygen. Okay, and as we already talked about, in the arteries, the partial pressure of oxygen is closer to 100 millimeters of mercury. In the veins, it's about 40. Okay, so this is the partial pressure of oxygen in the blood. On the y-axis, this is hemoglobin saturation. Okay, so what that means is this is how much hemoglobin is bound, or I'm sorry, how much oxygen is bound to hemoglobin. If every single binding site on the hemoglobin molecule has an oxygen attached to it, then you would be at 100%, right? Down here at 0%, this is actually where there is no oxygen bound to hemoglobin. All of the binding sites are empty. Okay, so this is the hemoglobin saturation, and it's in a percentage. So when you get up to the arterial blood and you have 100 millimeters of mercury of oxygen surging in that area, you can see that it's at about 100%. All of the oxygen binding sites have an oxygen molecule attached to it. But if you toggle down to what's happening in the veins, right, at 40 millimeters of mercury, you can see that only 75% of the oxygen binding sites actually have oxygen attached. Okay, so when we say deoxygenated blood, there's still a lot of oxygen bound to hemoglobin, 75%. So it's not completely void of oxygen molecules. It's only depleted about 25%, right? This is about 75% in the veins. Okay, so does everybody feel pretty comfortable about reading this graph? Is there any confusion or questions? Okay, so far so good, right? All right, so let's talk about what P50 means. What is P50? And how to measure it? It's basically from the point at which 50% of oxygen saturation is attained. So you would go to the 50% mark, you would go to the line, just read over to the line, and then go straight down. Read down, and that would be the required O2 partial pressure, known as the P50. Okay, so that's how to read P50. So if you actually had, let's go back to this one. What would the P50 be in this case? Go to the 50% mark, read over to the line, and then go down. In this case, the P50 would be about 25 millimeters of mercury. You guys with me on that? That's going to come into play here in just a second too. So, all right. Okay, so now it gets a little sticky. Now we're going to talk about some of the effects of carbon dioxide, acidity, and in this case, a molecule called DPG, right? Diphosphoglycerate. And you can just know it as, it's actually 2,3 DPG. Now, DPG is upregulated in your body at high altitude and when you're pregnant for women. Okay, so it's an interesting story with DPG. High altitude and pregnancy increases surging levels of DPG. All right, so let's take a look and see what happens. We're going to talk about these different, this is in your textbook, this is 1329, but I'm actually going to lecture from this particular graph. It's a little bit easier to see. So we're going to start, before we get too far down the road, this effect is called the Bohr effect. When you get this shift in the curve because of increasing acidity, carbon dioxide, and temperature, and DPG. This is all called the Bohr effect. All right, so what does it mean? Start off with pH. As you can see, with higher and higher levels of acidity, remember as the pH gets lower, the body gets more acidic, right? So as you increase the acidity, you can see that there is a shift of the P50 to higher levels. Here's the first one at high pH or lower acidity, and then you shift it to the right as the body gets more acidic, and you shift even more to the right when now the pH is very acidic at 7.2. Acidity causes the curve to shift to the right. Okay, what does that mean? Why is that important? What it tends to do, and you can write this down, I'm going to say this very slowly. An increase in acidity helps facilitate the unloading of oxygen. It gives up oxygen easier as your body becomes more acidic, right? Makes sense?
Say that one more time.
Yes, this is a really important concept. As you get a shift to the right, now in this case, as you increase the acidity in your body, you increase the P50. I'm going to go up to here to the P50. P50 increases, and it facilitates the unloading of oxygen. in environments that have higher acidity. It gives up its oxygen molecule easier. Does that make sense? Gives it up. So how do we know this? Let's take a look at this same graph in a little bit different perspective. Let's just take it at the veins, 40 millimeters of mercury. Okay? If we go straight up, right? Go straight up, you can see at the same levels of oxygen, the body, when it has lower acidity, has only given up about 83%. It still has 83% of the oxygen molecules attached. It's only given up about 17%, right? Because we're right here. 83% of those oxygen binding sites still have oxygen attached. It's really only given up about 17%. Now, as the body becomes more acidic, you can see, let's go down a little bit more. The body's more acidic. Now it's given up about 28%, right? We still have about 72% of those binding sites still have oxygen, but now the hemoglobin molecule has given up 28% with higher acidity. Now let's go to the highest acidity, 7.2. You can actually see at the very same oxygen levels, 40 millimeters of mercury, at that higher acidity, it's given up 40%. Okay, it's given up a lot more oxygen. So what that means, this again is called the Bohr effect. It's a phenomenon that says in higher acidity, the hemoglobin molecule tends to give up its oxygen molecules easier. You guys pretty good with this so far? I'm going to give you a way to think about this in just a second. All right, the same thing with carbon dioxide levels. As you increase carbon dioxide levels, you can see that it profoundly shifts P50 to the right. And same thing, right? At 40 millimeters of mercury, at higher CO2 levels, this is the highest CO2 level here at 80 millimeters of mercury, partial pressure of carbon dioxide. 50% of those hemoglobin binding, the oxygen binding sites, have given up their oxygen molecule. So the same thing, at high carbon dioxide, an environment that has high carbon dioxide, it tends to give up its oxygen molecules that are attached to hemoglobin easier. So another way of saying that is the affinity for oxygen decreases as pH decreases, that's higher acidity, or an increase in carbon dioxide levels. Yes, you have a question. Yes, that's a good point too, right? We've already learned that in the blood, when you have high carbon dioxide levels, you should have higher acidity in the blood. That's a good observation, yeah, good connection. Okay, so hopefully you guys can read the graph. Again, as you increase carbon dioxide, you increase P50 values. It goes from, let's say here, 15 maybe, to 40 millimeters of mercury. Remember, you have to read down. And you can see here, if you pick one particular partial pressure of oxygen at 40 millimeters of mercury, you can really see the differences. As you increase, I'm going to take my arrow here, as you increase carbon dioxide levels, it really facilitates the unloading of oxygen. The affinity for oxygen decreases as you increase those carbon dioxide levels. All right, here's one too, temperature, same thing. As you increase the temperature, it shifts the curve to the right. which facilitates the unloading of oxygen in that area. It decreases oxygen affinity and increases the unloading. Yes?
So if like hemoglobin is still holding on to a lot of its oxygen, how is the partial pressure of oxygen like the same?
So this is a really good question. Okay, so I want to make sure that you guys understand this question. Remember, hemoglobin, the oxygen that is bound to hemoglobin, doesn't play a role in the partial pressure of oxygen. Okay? So what we're talking about here is really the partial pressure of oxygen, which is the dissolved oxygen in the arteries and in the veins. Okay, so that's separate, right, from the oxygen that is attached to hemoglobin. It does have an effect, a kind of like a conformational effect on the hemoglobin, which lowers the affinity for oxygen and facilitates the unloading. It gives up, the hemoglobin molecule gives up the oxygen easier in higher acidity, higher carbon dioxide levels, and higher temperature. Okay, so before we move on to DPG, I want to just make sure that you guys think about this and apply it. Okay? So I always, when I think about the Bohr effect, I always think about exercise. Okay, so what happens with exercise, right? If you're running 3 miles and you know your quads start to scream, right? Because they're really working hard. What's going to happen is in the area, right, in those capillaries that are perfusing the muscle cells in your legs, you're going to get high acidity. That lactic acid is going to go up. You're going to have high temperature, and carbon dioxide levels are really going to elevate, right? That's all going to go up. So imagine a hemoglobin molecule traveling from the lungs to the quads, that area of quads. When you're exercising, what it does is that it really facilitates the unloading of oxygen. You got to deliver more oxygen to that area pretty quickly. So the environment changes, right, when you're exercising. High acidity, high temperature, high carbon dioxide. It helps facilitate the unloading of oxygen in that area. Does that help you guys? That helps you kind of remember which way, what's important in the shift to the right. High acidity, high temperature, high carbon dioxide. Okay, so this actually, typically you can see how that's really beneficial when you're looking at this shift to the right. The A is the shift in the arteries. Right? This is the hemoglobin oxygen dissociation curve in the arteries. And in the veins, because of the higher carbon dioxide and higher acidity, you can see it shifts the curve to the right. Okay? Now, if we only look at the arterial curve, you can see that when we toggle between the arteries, which should be more like 100 millimeters of mercury and 40, there's only a very small depletion. We're only looking at this curve right here. If we go from 100% to 75%, you can see that there's not much oxygen that's delivered. In the veins, you can see there's a little bit more oxygen, 6.5 if we go from 100 to 75. But if we're actually toggling between the arteries and the veins, the shift between the arteries and veins is a lot more oxygen delivery. So oxygen delivery to the tissue is greater in the presence of the Bohr shift, that Bohr effect between A and V, which is arteries and veins. So you're able to deliver more blood with the Bohr effect. Okay, this is just blowing up the same graph. You can take a look at this at home. I like to kind of talk about it with the balloons. It helps you read while I'm describing it. Again, the Bohr effect typically enhances oxygen delivery in animals. All right, so now let's talk about DPG. DPG is again upregulated, and it's produced in the red blood cells. It's upregulated in if you're at high altitude or pregnancy. Now, this one's a little bit harder to read. Right now, the y-axis is P50. Remember what P50 is, right? You can see with increasing levels of DPG, diphosphoglycerate, you increase P50, right? P50. So what that means, going back, remember if we take a look at one of those other graphs, here's the P50. As you increase P50, you're actually shifting the curve to the right. That's an increase in P50. Increase P50, it's a shift to the right, which facilitates the unloading of oxygen. So don't get too confused. As you increase P50, you can imagine this is just a shift to the right. Okay, so if you're at high altitude, this helps to facilitate the unloading of oxygen to your tissue. That makes sense. But how does it relate to the fetus? I love this story. Just remember that postnatal animals have 2 alpha and 2 beta, but fetal globin actually has 2 alpha and 2 gamma. DPG can only bind to beta globin. Now you might see the story already emerging. If it can only bind to the beta globin, that means that it facilitates the unloading of oxygen with the maternal, the mother's blood, but not the baby's blood. So now they don't compete, right? Now the baby can accept the oxygen molecules from the mother, right, without competition. The mother actually, because of the Bohr effect, releases the oxygen molecules a lot easier. The fetal globin has a much higher affinity for oxygen. So it's a very nice transfer of oxygen from the mother's hemoglobin to the fetal hemoglobin. Pretty good, you guys? So this is just some interesting information. This is actually giving you some information about the placenta and the umbilical cord. You can actually see that these fetal blood supply, which is these loops right here, is actually bathed with maternal blood. So this area that's surrounding these particular loops, there's a nice exchange between the mother's hemoglobin and the baby's hemoglobin. Yes?
Does this have anything to do with like infant hemolytic disease? Like, is this the spot where it happens?
Yeah, no, a lot of times that happens postnatally, and it has a lot to do with vitamin K, right? And so that's why, well, they used to, I don't know if they still do this, they actually give babies a shot right when they're born, right into their heel, to prevent that particular hemolytic disease. Yep. All right. So you don't really need to know the anatomy. If we were, talking about reproduction, that'd be one thing. All I want you to know is how DPG plays a role in the transfer of oxygen from maternal hemoglobin to fetal hemoglobin. Okay. All right, you guys. Let's see. Let's go to this one. This is one more. This is called the root effect. Now we're moving away from the Bohr effect and we're talking about what's called the root effect. In some species like eels, this saturation point, right? This is the hemoglobin dissociation curve. It actually is lowered when you have an increase in acidity. So how do you think about that? This saturation point is the total oxygen carrying capacity. So instead of just a shift to the right, it actually lowers the oxygen carrying capacity of hemoglobin. This is called the root effect. Okay. Now, I'm going to talk about the Haldane effect and carbon dioxide transport on Friday. We still have about 10 minutes, you guys. I don't want to overload you. Oxygen dissociation curves are very complicated. If you're still a little fuzzy about it, I can still, I'll stick around and talk to you about it. What I would suggest in the last 10 minutes, I'm going to go easy on you guys right now, is if you have any questions about your worksheets and you want to finish up some of the problems and turn them in, you can definitely do that. I'll walk around. Also, what I'd like to do is just show you the severe dehydration, right? Let's see, get a new window here. We'll go to our Moodle website real quick. And I will post this. We've already done this worksheet. And I will also post the answers to the essential respiratory calculations and the into thin air. But let me show you the answer for the severe dehydration. I meant to do this the other day. There's only one thing that I want to mention. All right, let's see here. Okay. All right, let's see if we can zoom in on this. It's pretty crazy. All right. Okay. It probably looks similar. I'm not going to, don't get too caught up. It's actually a little bit similar. Okay, so what do we have here? Remember, it's those hypothalamic osmoreceptors that are going to detect the increase in osmolarity within the blood. Does two things. It initiates a profound thirst behavior, and it releases vasopressin. Remember, ADH is also antidiuretic hormone, which is vasopressin, from the posterior pituitary. All of this, the increase in water intake and the increase in water reabsorption, right? This is actually an antidiuretic. It helps you transport water from the nephron back into the blood, helps to increase blood volume, and I would say increase mean arterial pressure. One more box, increase mean arterial pressure. Now, on the flip side here, the middle one, when the low blood pressure and blood volume is detected by the carotid and aortic baroreceptors, that sends a signal to the cardiovascular control center to increase sympathetic tone, which increases heart rate, stroke volume, and vasoconstriction, right? That actually has an effect on an increase in cardiac output, total peripheral resistance, and finally mean arterial pressure. And then on this side, this should be the renin-angiotensin system. Those juxtaglomerular cells are going to detect the decreased flow at the macula densa, release renin. Angiotensinogen is going to be converted to angiotensin 1. ACE converts 1 to 2. And you're going to get vasoconstriction, right? That results in vasoconstriction, an increase in total peripheral resistance, and an increase in blood pressure. But this is interesting. You probably said that you get an increase in aldosterone, and I wouldn't have expected this from you. You're not going to get this wrong. This was just participation. But instead of an increase in aldosterone, the hypothalamus overrides and decreases aldosterone, only because it doesn't make sense to absorb more salt in the face of already high osmolarity in the blood. Okay, so I bet yours is pretty close. I know this is intimidating, but I bet you did a pretty good job. Okay, all right, so again, the review session is at 2 o'clock. I'm going to stick around for any questions. You don't have to stay, but if you have any last minute questions on any of your worksheets, I'll be around. All right.
Hi.
Hi.